Hello and welcome to this edition of the Redtalks. It's my honor to be here. My name is Mark Reshevsky. I'm a clinical investigator, and as my background tells you, I work in the United States at the National Cancer Institute here in Bethesda, Maryland, where, of course, we have a strong research interest in understanding both the biology and therapeutic weaknesses of a variety of B-cell lymphomas. Today, we're to talk about follicular lymphoma. And it's my pleasure to introduce our speaker, the distinguished Professor Oliver Weigert. So Dr. Weigert graduated from medical school at the Technical University of Munich in 2002. He then obtained his medical degree, his MD, in 2004. He then completed both his residency and fellowship training in internal medicine at the Ludwig-Maximilians-University Hospital. He was board certified in internal medicine in 2009 and both hematology and medical oncology in 2015. From 2009 to 2012, he was funded by the German Research Foundation and then joined Professor David Weinstock's laboratory at Dana-Farber Cancer Institute, otherwise known as Harvard Medical School in Boston, as a postdoc research fellow. There he studied genetic resistance mechanisms in a variety of lymphoid malignancies and the molecular ontogeny in follicular lymphoma. Currently, Professor Weigert is a senior attending physician in the Department of Hematology and Medical Oncology at the LMU Hospital in Munich, where he co-heads the clinical lymphoma program and was appointed assistant professor in 2019 and full professor for translational lymphoma research in 2021. He currently co-heads the Laboratory for Experimental Leukemia and Lymphoma Research and the core facilities digital molecular barcoding and patient-derived organoids of the German Cancer Consortium. Furthermore, Professor Weigert served as an elected spokesperson of the Biological Research Working Group of the German Lymphoma Alliance from 2017 to 2021 and has been a member of the GLA Board of Directors since 2021. So Dr. Weigert's laboratory uses agnostic multi-omics approaches optimized to comprehensively profile formalin fixed paraffin-embedded primary patient material, currently including over 1,000 follicular lymphoma biopsies from clinical trials. Some of the major seminal findings have included the development and validation of clinical genetic risk models, such as the M7-FLIPI, published in 2015 in Lancet Oncology, and the POD24 PI, also published in 2016 in Blood. Furthermore, they use advanced in both in vitro and in vivo model systems for functional characterization of clinically relevant candidate alterations, such as cathepsin S and STAT6, also led to seminal publications. So during this talk, Professor Weigert will try to describe the evolution of biomarkers that we use in follicular lymphoma and how he envisions us gaining a deeper understanding of the underlying biology in order to ultimately drive biology-guided treatment decisions in follicular lymphoma. So without further ado, Professor Weigert.
Thank you very much, Mark. I feel truly honored to be allowed to give this presentation. And this was essentially my complete bio sketch. Usually I'm used to introduce myself, but this was very granular, and thank you very much. This was very kind of you. I'm not only honored to be here, I have to admit I'm also tired. I did attend, as many of you, I did attend ASH. It was a really long commute. So my apologies if I'm a little slow. I think I shared my screen now. Mark, can you quickly confirm if you have my slides in presentation mode?
Yes, we can see your slides.
Because from now on, I only see my slides and nothing else. All right, so the flight was really long. I didn't intend to spend too much money on my flights. I was not funded by industry, which is fine. But these are my disclosures. This is what I'm being funded for, essentially research funding, which I'm grateful for. Here's another disclosure. My talk will be very biased. So it's going to be my opinion. It's going to be primarily our work. And I do apologize to my colleagues. I will not be able to include much of their excellent work in today's presentation, simply because I'm not qualified enough. This will be today's agenda. I like to take the opportunity to clarify some of the, I think, conceptual misperceptions on the POD24. I try to place our work on biomarkers in context. I try to outline how I think we can transition from descriptomics to biology. And I will also share some unpublished data. I think that's the spice of this kind of presentations. This is definitely not needed for this audience, but let me quickly remind you, I will only talk about follicular lymphoma, and I will actually only talk about classic follicular lymphoma. You know, we have two classifications these days. So this is nodal classic follicular lymphoma. And as you know, this type of lymphoma is among the most common lymphomas worldwide. It's considered the prototype of indolent lymphoma. You will later hear that this is a term that I don't like very much. As you know, many of these patients are being diagnosed at advanced stage disease, and we still consider these patients to be incurable. Essentially, if these patients require treatment, if they have symptomatic disease, they get CD20 antibody, first-line treatment, usually combined with chemotherapy, essentially irrespective of individual risk factors, essentially irrespective of disease biology, although we know that follicular lymphoma is a heterogeneous disease, both clinically and molecularly. You have seen this data a lot, and this data is essentially used to emphasize the fact that follicular lymphoma is a clinically heterogeneous disease. So it all started with beautiful work from Carla Casulu, looking essentially at patients and distinguishing these patients, whether they had early progression of disease or not. And as you can see on the left side, patients with early progression of disease had dismal overall survival, as you can see here. And early progression of disease in this analysis was essentially defined as having progression of disease within 2 years. So back then we set out to validate this data, and this is shown on the right. And we used some slight modifications. So number one, we introduced the term POD24 because we felt that early progression of disease is somehow vague. So we wanted to remind people that this is essentially 24 months. And we used time of initiation of treatment as the start point, unlike Carla Casulu, and because they essentially used initial diagnosis as their starting point. So I think most people have now used our definition starting at time of treatment initiation, and also used the term POD24. So 24 months was essentially established by the Mayo Group because it was the ideal time point to distinguish patients by their treatment outcome in the context of R-CHOP treatment. So essentially all of these patients received R-CHOP as their initial treatment. And so did we. But I want to remind you that treatment matters, right? And the more effective your treatment, the lower your POD24 rates will be. So for instance, if we use modern immunochemotherapy, as we did in the Gallium trial, using a more effective CD20 antibody, now using anti-CD20 antibody maintenance treatment, as established by the French group, by the PRIMA trial, the POD24 rates drop. So like initially we said like it's around 20-25% of patients who have POD24. Within the gallium trial, it was less than 10%. It was actually 7%. So treatment matters. And like if you were to treat patients with rituximab monotherapy, like your fraction of patients with POD24 will be higher. Ideally, for every type of treatment, we would define the optimal cut point. But I see this is not being done these days. So people simply report a POD24 in every context. And I think that's problematic. Number two, the initial idea was that patients with, I think this was what people perceived, that patients who are not having POD24, the non-POD24 patients, have less aggressive, more indolent disease. And again, I think this concept does not hold true because many of these patients in the non-POD24 group will have excellent outcomes, but patients will also have relapses and progression. And we, like, in the interest of time, I will not show the data. But if you look at these patients with progression of disease beyond 24 months, they are actually patients who have poor outcome. So I think it's not necessarily true that non-POD24 patients have more indolent clinical course. So I just wanted to make that misperception clear. What I really agree with is that patients who have early progression of disease, for instance, within 24 months after standard immunochemotherapy, they have more aggressive, they have more problematic and clinically challenging disease. And the question is, how do we deal with that? And this is analysis that we did a couple of years ago. And as you can see, and as others have been reported as well, high-dose therapy and autologous stem cell transplantation is an option for these patients. So we're comparing the blue and the yellow curves. And as you can see, in a subset of patients who qualify for those intensified treatment regimens, they benefit in terms of longer progression-free and also overall survival. And that's actually still our recommendation in Germany, and I guess in many other countries, that patients with early progression of disease who qualify for autologous stem cell transplant should be offered that type of treatment. So who does qualify? Obviously, patients who are fit and patients who have chemosensitive disease. And this will be your experience as well. This is not a lot of patients, and having now all these new treatment options, it's actually fewer and fewer patients whom I actually consider eligible for this approach. Those of you who have attended ASH this year, last year, etc., like there's exciting new treatment options, right? Novel treatment, novel compounds, T-cell-based therapies, CAR T-cells, BiTEs, etc. They're actually moving in earlier lines of treatments, including in clinical trials, first-line treatments. And the challenge is like how to identify the patients who are at risk of having inferior outcomes with our standard therapy. So whom should we prioritize to have these novel therapies early on, and how to identify subgroups who benefit from specific novel therapies. And I think unlike the many treatment options, we are running low on respective biomarkers. So before we get really started, let's quickly talk about biomarkers in general, because like sometimes I see that prognostic and predictive is being mixed up. So let's assume we have an effective treatment. So if we have a biomarker that is neither prognostic and predictive, you will still see a difference whether patients receive an effective treatment, but there will be no difference with respect to the absence or presence of the biomarker, right? So if a biomarker is purely prognostic, there obviously will be a difference, right? There will be a difference in patients who are not receiving effective treatment, and there will be a difference in patients who receive effective treatment. And those patients with the biomarker positive, and let's assume that's a biomarker associated with more favorable outcome, your progression-free overall survival, whatever, will be longer, but the delta will be similar. So let's so I think that's partially helpful, right? So you can tell the patient about his prognosis, but in this setting, there's nothing you can specifically do about it. So you can simply inform the patient. So that's helpful, but not ideal, right? So on the other hand, a biomarker can be purely predictive. And now that's actually clinically more useful, right? Because like this biomarker may not have any impact in the absence of treatment, but with a specific type of treatment, a particular treatment with a particular type of biomarker, these patients will actually have significantly superior outcome, right? So that's highly clinically useful. And again, there's a combination of both. So a biomarker can be both prognostic and predictive. And that again is highly useful in the clinic. So what we actually want is predictive biomarkers, at least to guide treatment decisions for our patients. So one last conceptual thought before we dive into the data is this is how I like to think about biomarkers. The more cases, the more patients, like the more data you actually have on individual patients, the more clinically useful, the more clinically robust your biomarker will be. And on the other side, like the more data items, the more layers, the more omics you actually have, the more biological insights, the more biologically relevant your biomarker will be. Right? So this is what we have been doing traditionally over the last decades or so, right? Like hundreds and thousands of patients were analyzed for the absence or presence of a particular biomarker. And here is like maybe the poster child, TP53 mutation status, clearly shown by the group of Jude Fitzgibbon and later confirmed by us and many others, presence of TP53 mutations clearly associated with inferior progression-free and also overall survival. Yet clinically of limited value for a variety of reasons, like number one, less than 5% of our patient initial diagnosis actually are TP53 mutated. And number two, I think it's a poster child example for a purely prognostic biomarker. So at least to the best of my knowledge, there's no good treatment strategy that we can specifically give to patients with TP53 mutated disease that will alter their disease risk. Second type of biomarkers, I'd say, are more modern biomarkers, again, integrating now more data, and we are in the range of hundreds to thousands of patients having been profiled. And this is early work, and I will, from our lab, and I will go quickly because this is fully published. We determined the mutational landscape of patients with follicular lymphoma. We integrated the mutation status of these genes with clinical risk factor and the performance status. We actually did a penalized Cox regression, which now many people would more modern coin as machine learning or AI technologies. That's essentially what we did. And what we ended up is the so-called M7-FLIPI, a score that integrates the mutation status of 7 genes, like simply absence or presence of the mutation. This is something we can, I'm happy to discuss. This has limitations. And these risk factors are being individually weighed, and you sum up the absence or presence of these factors, you have a cumulative score that we then dichotomize to distinguish high risk from low risk patients. And this is how it performed. So we were actually able to reclassify about half of the patients with high risk FLIPI. So this is one of the standard clinical risk tools to be actually low risk by M7-FLIPI. So half of the patients in this curve actually moved up to here. And this leaves us with a smaller cohort of patients that have worse outcome. And this was actually independently validated in another cohort. To sum this up, the M7-FLIPI is a prognostic biomarker for failure-free survival, progression-free survival, and overall survival. It also was predictive of POD24. in the context of R-CHOP-like treatment regimens, and that's important. So this was the treatment context where this score was established and validated. Of course, gene mutations are just one way to molecular characterize these tumors. For almost 20 years, we have known that the tumor microenvironment has prognostic value, and this is pivotal work done in Lou Stout's lab back then, published by Sanjeev Dave, the New England Journal. And early on, they identified what they called immune response signatures, and they were associated with treatment outcomes. Have to say that like this work specifically was never validated as predicted by Rob Tibshirani early on in a letter, but the concept was clear, and later you guys, the French group spearheaded by Gilles Salles and Sarah Huet, essentially took that concept, looked into RNA sequencing data, later used nanostring data in order to identify differentially expressed genes that were associated with progression-free survival, and they came up with this 23 gene expression score shown on the right. And a lot of these factors actually represent the tumor microenvironment. And this is how it performed. So to the left is the training cohort, to the right is a really big validation cohort. So essentially, if you compare the M7 FLIPI and this gene expression score, very similar performance, and I'd say like the only reason why the M7 FLIPI is predictive for overall survival and is the 23 gene expression score is not, is the fact that the M7 FLIPI includes the FLIPI clinical risk score, whereas the 23 gene expression score is purely based on gene expression profiles. So we had the opportunity to validate these scores in a more modern clinical trial. This was the Gallium trial. As you know, the Gallium trial was comparing two different antibodies, obinutuzumab versus rituximab, combined with a choice of 3 different chemotherapy regimens. It was either CHOP, or like very few patients received CVP, specifically in the UK, or bendamustine. So this was, so participating centers could actually choose which type of chemotherapy they wanted to give together with the CD20 antibody. And as you know, the major findings were like that obinutuzumab significantly prolonged progression-free survival, like essentially with all 3 chemotherapy regimens. So we had access to primary patient material and did target DNA sequencing. And to cut a long story short, and I will not show too much data on that, but we could, in fact, independently, again, validate that the M7 FLIPI was predictive of failure-free and progression-free survival. And it was predictive specifically in the in patients who received CHOP or CHOP-like, meaning CVP regimens. So the effect was essentially identical to what we initially observed, and it was so pronounced that it actually was detectable in the overall cohort. But this effect was limited to the patients with R-CHOP and R-CVP, and the M7 FLIPI was not predictive in patients who received immunochemotherapy based on the bendamustine, combined with bendamustine. So we looked into, so there's clearly an interaction between the chemotherapy and the performance of the M7 FLIPI. So we did an interaction analysis. It's essentially looking what type of mutation interacts with the chemotherapy. And it turns out that it was actually the EZH2 mutation status that significantly interacted with the efficacy of the chemotherapy. So in terms of Kaplan-Meier curves, this is what it essentially looks like. So if you have an EZH2 mutant follicular lymphoma, you are significantly more likely to do better with the CHOP-CVP regimen combined with either rituximab or obinutuzumab. So that's the blue curves compared to the red curves. And on the other hand, if you have an EZH2 wild-type follicular lymphoma, you are significantly more likely to do better with bendamustine as compared to CHOP-CVP. So that's the gray versus the red curves. And this is true again in both randomization arms. So I think that's really a promising predictive tool. So is the gene mutation status the only tool that interacts with chemotherapy backbone? No, it's not. It's actually also true for the 23 gene expression score. So this is work that we've done together with Roche. And as you can see here, the 23 gene expression score high patients were validated to do poor with CHOP-CVP, but they were the ones who benefited most from bendamustine. Whereas if you were low in this 23 gene expression score, like there was no difference between bendamustine and CHOP. So I think, again, an interesting signal that might help us guide treatment and select the most efficient chemotherapy backbone for our patients. Let's quickly move to the more novel biomarkers, now actually integrating even more molecular data. This is something that I was not directly involved in, I just want to mention it. So from the UK, actually a larger DNA NGS panel that they did, they profiled a lot of patients, more than 500 patients, and they essentially distinguished 3 different groups by unsupervised clustering. Follicular lymphomas that were mostly characterized by aberrant somatic hypermutation events. A second group that was mostly characterized by the presence of STAT6 mutations combined with CREB-PP mutations. Then a large group that did not have these features, but other characteristics, molecular characteristics of follicular lymphoma that we frequently find, such as KMT2D mutation, etc. Even more comprehensively, group of Ryan Moran, shown to the right, used whole genome sequencing, and their angle was a little different. So they compared whole genome sequencing data of follicular lymphomas to diffuse large B-cell lymphomas, and then also had like patients that were transformed and were not transformed. And essentially using whole genome sequencing, they came up with 2 subtypes of follicular lymphoma that they termed constrained follicular lymphoma or diffuse large B-cell lymphoma like follicular lymphomas. And in that paper, they actually tried to facilitate, like you don't necessarily need whole genome sequencing, but you can actually use fewer markers that will give you a very high likelihood of distinguishing these 2 subtypes. And most prominently, it was like the head domain mutations in CREB-PP that were, and mTOR gene mutations that were significantly associated or made it more likely to be associated with constrained follicular lymphomas, and BCS6 transcription start site mutations, GNA13, etc., were more likely to be associated with the diffuse large B-cell lymphoma, like follicular lymphoma. Like this is a quote from their manuscript. This being interesting right now by itself in terms of biology, there was actually limited information to predict individual patient risk. Okay, and now the next level, and this is where we're heading at, is integrating more layers of information. I think this will be the future. Something that we are involved in, but that is not ready to be shared yet, is like having not only gene mutation data, not only having gene expression data, but we also have copy number, we have translocation status, methylome, etc. And all of that is being integrated in one way or another. Right now, we are mostly working with the non-negative matrix factorization, which has been successfully applied to diffuse large B-cell lymphomas. But I think there's a lot of effort going on in that direction right now. So we teamed up with colleagues from UK, Jessica Ockerson, and from Canada, Robert Kreidel, to have a large clinically meaningful cohort. We've seen a lot of spatial and single cell data at ASH. I think this is also something that will be that we will see in the near future. There was beautiful work from Bertrand Nadel's group. Here I'm showing work from Sarah Hebe from Stanford. This is an additional layer that I think will be very informative. And finally, again, a lot of that has been shown at ASH. I think dynamic monitoring can be a real game changer. Our moderator, Mark, NIH, but also the Stanford group is really spearheading this field. Like here, I'm showing data from diffuse large B-cell lymphoma from David Kurtz and colleagues. But again, there's been beautiful work shown, again, by the Stanford group, but also by others, that we will be able to integrate ctDNA and subsequently also PET-CT data to assess risk in our patients. That being said, ctDNA is a little more challenging in follicular lymphoma due to lower levels as compared to diffuse large B-cell lymphoma and also mantle cell lymphoma. All right, so that's the brief summary of biomarkers. I think this is where the field is moving to, but I still have to say that, like, this is still descriptive data. This is association studies. This is what I call descriptomics. So I think what we still need, despite the very high granularity of the data, is we need insights into the biology. And this is where I think the field will also move into, and this will be highly informative for future strategies. So I've told you that early treatment failure is highly significantly associated with short survival. I told you that cell intrinsic alterations, somatic alterations, etc., are informative, and I told you that features of the tumor microenvironment determine outcome, and what my lab and others, including Karin Tarz, etc., are interested in, like, what's the interplay? So, like, how do these two things come together, right? So I'm not the expert talking about the tumor microenvironment. Karin Tarz would do a much better job than I can, but I will tell you that follicular lymphoma depends on the tumor microenvironment, hijacks a lot of physiologic pathways within the tumor microenvironment. Actually, also this is a term that has been coined by Christian Steidl, re-educates the microenvironment, receives the necessary supportive signal, and at the same time avoids destruction, right? The anti-tumor responses that are also present at the same time. So my lab is interested in better understanding the impact of certain gene mutations or patterns of gene mutations and how they impact the interaction, the crosstalk, the bidirectional crosstalk between lymphoma cells and the different components of the tumor microenvironment, mostly the cellular components, but there's also non-cellular components of the tumor microenvironment that are relevant in that aspect. So the first one I want to quickly summarize, STAT6 mutation. We are standing on shoulders of giants here. Karin Tarz has beautifully demonstrated the importance of IL-4. IL-4 derived from T follicular helper cells, actually a particular type of T follicular helper cells. IL-4 binds to the heterodimeric IL-4 receptor, activates the JAK-STAT signaling pathway. And then, like, patients with STAT6 mutations have increased, augmented STAT6 signaling activity. And contribution from our lab actually was that mutant STAT6 specifically binds to the promoter of PARP14, and PARP14 happens to be a component of the enhanceosome complex of STAT6. So further stabilizing this complex, having increased transcription activity for STAT6 target genes, and this will result in expression of STAT6 target genes, and this includes chemokines and cytokines that are again implicated in re-education of the tumor microenvironment. And again, I think this is an opportunity for specifically targeting these pathways. So like we've demonstrated that PARP inhibition is actually able to counteract this phenotype. We've also demonstrated that the STAT6 mutations by themselves have no gain of function activity, but they really need activation signal from upstream. So if there's no IL-4, if there's no JAK-STAT activation at this point, these gain of function mutations have no phenotype. So also inhibiting upstream of STAT6 in this case is a promising treatment option for this defined subset of follicular lymphoma. Second example, again, I will go quickly, are cathepsin S mutations. Cathepsin S is a lysosomal cysteine protease and work spearheaded by Johannes Hildebrand in my lab actually demonstrated that there's CTSS hyperactivity in a subset of follicular lymphomas. And this hyperactivity is either mediated by gain of function mutation. So there's a hotspot mutation that actually allows CTSS, cathepsin S, to be activated more efficiently. So it from its inactive preform to the active enzyme. And another set of lymphomas actually have CTSS amplifications. So both of that results in cathepsin S hyperactivity. And this was actually published back-to-back with Elisa Auricchio's group, highly similar and confirming data. What cathepsin S hyperactivity does within the lysosome, it actually increases antigen presentation. It actually also cleaves CD74. It releases CLIP, so essentially allows MHCs to be allows antigens to be loaded on MHC class 2. This results in more efficient activation of T cells, CD4 T cells, and this creates a tumor supportive environment. And again, like this is I think a wonderful example that like targeting the tumor promoting tumor microenvironment in defined subsets of follicular lymphoma might be a promising treatment approach. So all of this was published. Now let's dive into unpublished data. I want to talk to you about the impact of ARID1A mutations in follicular lymphoma. So why study ARID1A mutations in follicular lymphoma? So it all started during my postdoc. So back then, we described a case of donor-derived follicular lymphomas. So essentially, after an allogeneic bone marrow transplantation, both the donor and the recipient developed follicular lymphoma. And we could show that the common precursor cell was actually identical. So these lymphomas were actually arising from the same precursor cell. And a lot of the mutations were actually shared and detected early on. But ARID1A mutations were not shared, yet we were still able to identify disruptive ARID1A mutations both in the donor and the recipient. So they were acquired independently in both lymphomas. And back then, this convergent evolution already suggested functional relevance. And I think this was the first description of an ARID1A mutation in follicular lymphoma. And afterwards, with all the targeted sequencing data being available, like we identified and others identified that ARID1A mutations are really highly recurrent in follicular lymphoma, and again, mostly disruptive. And finally, you may have seen and noticed that ARID1A mutations were also part of our M7 FLIPI score. So the presence of an ARID1A mutation was actually lowering the risk of having early treatment failure. So both the recurrence, the presumed functional relevance, and and the fact that it's obviously clinically relevant made us look deeper into the role of ARID1A mutations in follicular lymphoma. So what ARID1A actually is, it's part of this multi-mer SWI/SNF nucleosome remodeling complex. And what it actually does, it loosens the interaction between nucleosomes and DNA. And actually, then the nucleosome can slide along the DNA, and actually nucleosomes can also be ejected. So what this complex actually controls is the openness of DNA, right? And thereby controlling a lot of processes, including gene expression, but also DNA repair, etc. So this is the reason why we decided to study ARID1A. So these ARID1A mutations result in ARID1A haplodeficiency, like we've seen that early on. So here you see immunohistochemistry stain of a wild type lymph node. This brown stain is actually ARID1A. So if you have copy number loss or the nonsense mutation, as you can see, there's less brown staining. So these mutations are not only disruptive, but they result in haplodeficiency of ARID1A. And this can also be appreciated on the Western blot. So if we have ARID1A wild type cells compared to ARID1A mutant cells, like there's significantly reduced protein expression in the mutant cells. We were intrigued by reports back then identifying ARID1A as a regulator of FAS. So this was actually an RNAi screen done in a variety of cancer cell lines, and they demonstrated that disruption of ARID1A actually mediated resistance to FAS ligand-induced apoptosis. So this was something that was really intriguing. The reason for that being that, as you know, during the pathogenesis of follicular lymphoma, you have this iterative rounds of germinal center transition. Within the dark zone, you have this highly proliferative milieu. And in the light zone, there's a crucial process going on. It's negative selection of B cells. And physiologically, autoreactive B cells or potentially malignant B cells are being counter-selected, are being negatively selected, removed. More than 99% of our B cells actually die within the germinal center reaction. And this process is mostly driven by FAS. So losing FAS, we hypothesized, would give these cells a tremendous selective advantage. So this is the cohort that we again analyzed, 151 patients from the GLSG 2000 cohort with available gene mutation data. So we had 124 cases being wild type, another cohort being mutant. And as you can see, the mutation profile as expected, mostly truncating disruptive mutations. Also mutation frequency as expected. And then we did gene expression profiling. And then we saw, I'd say, a trend towards lower FAS expression. Now you have to know that FAS is also highly expressed in other immune cells, especially T cells. So what we really needed was something with single cell resolution. And we teamed up with Martina Rodelius, our skilled pathologist, and she did quantitative multispectral imaging. And as you can see, if we then really zoom in the follicular lymphoma cell population, the mutant lymphoma cells have significantly lower FAS expression. So we then, for functional studies, moved to cell lines. Again, we confirmed these two gray bars represent wild type cells, the two blue examples for mutant cells, and as you can see, significantly lower FAS expression, although to a different extent. For functional studies, we actually wanted to have comparable conditions. So we used the two wild type cells, OCLI1 and OCLI8, performed CRISPR to generate clones that had heterozygous or loss or complete knockout of ARID1A. We actually were also able to rescue ARID1A expression, which is difficult because this is really a large protein to work with, 250 kilodalton. And again, we confirmed lower FAS expression in het and knockout clones that we could actually rescue with ARID1A. And this is FAX, so essentially assessing cell surface levels of FAS. And we confirmed this by mass spec. And we also see differences in total protein levels depending on the genotype. So decreasing loss with decreasing loss of ARID1A. Again, something that can be rescued with the re-expression of ARID1A. So remember this Weiss-Liff complex and its function. So we obviously hypothesized this is reduced gene expression, and we performed RNA-seq and validation by qPCR. So as expected, mutant cell lines indeed had lower gene expression. And again, we hypothesized, and this was also the hypothesis in the literature, that it will be the promoter region, right? So this is what this Weiss-Liff complex does. It controls the openness. So we tried to confirm this performing ATAC-seq, and to our surprise, there were no differences. So we actually saw what we expected. So we teamed up with two bioinformaticians, Maria Solovey and Maria Colomitachi, and saw no differences at the FAS locus in neither of the cell lines. So it's not the differential openness at the promoter that results in differences in gene expression and protein expression of FAS. So then we said, like, if it's not the promoter directly, it must be some FAS regulating transcription factors. Again, like we searched the known FAS and predicted FAS transcription factors, checked for their openness, and they intersect with gene expression data. But again, this was not the explanation. There were no mutually affected FAS transcription factors detectable that would explain that phenotype. So then the next level was, let's check the co-transcription factors. And indeed, this analysis yielded a hit. We identified ETS1 as a predicted FAS regulating transcription factors. And ETS1 was predicted to cooperate with RUNX3 simply based by homology. So this is what it actually looks like. Here is the ETS1 locus, right? No differences. And here's the RUNX3 locus and differences in openness. This is what the RNA-seq data looks like. ETS1 matching this ATAC-seq profile, not differentially expressed, but RUNX3 significantly reduced expression in the presence of an ARID1A mutation. So we wanted to, like, all this was predicted, so we wanted to functionally validate this. So we first took a look at the genomic context. These are the regulatory elements of FAS. This is the promoter region. These are the ETS1 binding sites. In blue depicted the open peaks. This is actually ChIP-seq data available from the literature. And in this case, like, the overlap prompted us to clone this region that contained this transcription factor binding motif and performed ETS1 reporter assays. So we expressed increasing doses of ETS1, and for both constructs, we really saw increased transactivation activity. I'm not showing the data here, but when we expressed RUNX3, there was no transactivation activity at this promoter. And finally, we also confirmed that ETS1 and RUNX3 really directly interact. So this is an IP with ETS1, this is a Western for RUNX3, and as you can see, lower levels of RUNX3 and increased levels when we pull down wild type. Again, we performed rescue experiments. So as you can see here, the different genotypes for ARID1A, we re-expressed RUNX3 to high levels in both cell lines. And as you can see here, for the heterozygous clone, reduced FAS expression, this can be rescued with RUNX3, and in the knockout clone, this can also be partially rescued, and the same is true for another cell line. This is FAS gene expression, and this is FAS measured by flow cytometry in the cell surface. So why am I all telling this mechanism? Is it functionally relevant? This is what we did next. We performed, we exposed these tumor cells with and without the RNA mutation to FAS ligand. And as expected, so the black curve is the wild type cell line. If we have a heterozygous or a complete RNA knockout, these cells responded less well to FAS ligand induced apoptosis. In this case, the knockout was really resistant. Yet when we re-expressed RUNX3, and the same was actually true for L1A, we could rescue this phenotype. And again, this was true in both cell lines. Again, this is not how FAS is usually engaged. Usually FAS is membrane bound on T cells. This was the experiment we did next. So we exposed these different lymphoma genotypes to CD8 T cells. Importantly, we did not see any differences in conjugate formation. So this was not the reason for the difference we saw here. We could really attenuate killing in the heterozygous and the knockout clones, and by re-expression of RUNX3, we could rescue this phenotype, which leaves us with this model that in L1A wild type cells, you actually have an open RUNX3 promoter. RUNX3 cooperates with ETS1 to regulate FAS expression, and FAS actually then allows these cells to be killed by T cells. And in the presence of an L1A mutation, that promoter is closed, cannot cooperate with ETS1. You have fewer FAS on the cell surface, and so these cells can evade T cell mediated killing, which we hypothesize is a selective advantage during the German center reaction. Now, do I care as a clinician? I do. And this is the reason why. Like with all of the now available T cell based therapies, I think this is a relevant phenotype to keep in mind. Because as you know, like for instance, a CD19 CAR T cell will definitely bind via CD19 to our follicular lymphoma cells, but a lot of the killing will actually depend on FAS ligand interaction, and maybe less so for BiTEs, T cell engaging bispecific antibodies. But in the lab, if we use a T cell blocking, a FAS blocking antibody, we can also attenuate that effect. So I think FAS expression on the cell surface will make a difference. And this has been demonstrated before. So this is really nice work in a different context. So this was large cell lymphoma. And also this was differently assessed. This was ctDNA, and they did low pass whole genome sequencing from ctDNA. But like the top hit associated with inferior response to CAR T cells was really loss of FAS, which is on chromosome 10, right? And do I think this is relevant in follicular lymphoma? I also think it is particularly relevant in follicular lymphoma because, like, we and others have cohorts that span the evolution of follicular lymphoma, and what we have not yet published, but what we see is an increase in the frequency of disruptive R1A mutations from low to advanced stage, and a further increase in relapsed refractory disease. So having this immune evasive phenotype will probably, is my hypothesis, matter in the context of CAR T-cell therapies, and will have to be taken into account when we think about the timing when to apply these therapies. So this is the summary. I told you about my concept in how to think about biomarkers in follicular lymphoma. I think this is where the field is moving. The more data we have, the more patients we have, the higher the clinical and the biological relevance. But this will not keep us from doing functional studies if we want to move from risk-adaptive treatments to biology-guided therapies. So with that, I think I'm done. I have to thank a lot of people. I have a wonderful team. I have wonderful collaborators. I'm very grateful to the funding I receive, and again, the environment is important. I have a wonderful environment also with my co-workers. And with that, I thank you a lot for your attention, and I'm looking forward to our discussion.
Thank you so much, Oliver, for this very inspiring talk. Very, very interesting. It was very interesting for, I think, younger and less younger people to see also all the discussion on the microenvironment and the biomarkers. I like a lot your last slide for sure. Now it's time for question. I don't see if we have some question for the room, but perhaps I could start with one question. So about your last story about ARID1A mutation, do you see any difference in the patients that are mutated or not for the expression of MHC molecule on the surface of the tumor cells? Do you think that being resistant to fast targeting could allow them to retain MHC expression, and that it could give some sensitivity to other kind of therapeutic approaches?
Yeah, so this is actually what we checked, and we have not detected that. So we do not see in our model systems and in the not so many lymphomas that we analyzed, we have not seen difference in MHC expression, both MHC class 1 and 2. And this is also why I showed the data that the conjugate formation in our model system was not different. So if we have alterations, prime example being EZH2 mutations, they reduce MHC expression, then we see fewer conjugates. This is not what we see in ARID1A mutations. There has been a lot of wonderful data actually presented at ASH by Ari Melnik's group on ARID1A mutations and others. So that's right now a topic of interest, I'd say. Yeah. But like we don't see differences in MHC expression right now.
Okay, thank you. Perhaps another question for me. So as you know, I am quite interested by the TSH and IL-4 stuff. So do you think really you could think about targeting IL-4 receptor pathway? Because now there is some drugs to do that, not in lymphoma, but in other settings. Do you think there is a possibility, there is a plan for this treatment in follicular lymphoma?
So I think there's a possibility. As you know, there is the IL-4 antibody used in a different clinical context, but it's available, it's approved, it's available. Same is true for JAK inhibitors, right? We use JAK inhibitors quite a lot. So I think so how do I say? So for BTK inhibitors, for instance, they work if you have upstream activation events. And if you use a BTK inhibitor and you have an alteration that activates the B-cell receptor pathway downstream of BTK, they are no longer active. This has beautifully been shown by Lou Stout and co-workers, right? I think this is not true for STAT6, because the STAT6 mutations, as I tried to point out, are not gain of function by themselves. I think the original publication implied that. This is not what we see. And like, we tried a lot and hard. So they by themselves are not gain of function. They require either stimulation by IL-4. There's IL-4 activating mutations, IL-4 receptor activating mutations, beautifully shown by Christian Steidel's group. It's the same principle. So you have upstream activation, and this is required for the gain of function phenotype of STAT6. So yes, I think it makes sense biologically. Now here's the caveat. These STAT6 mutations are frequently found in limited stage lymphomas. They are frequently found in the absence of the 14/18 translocation. There's actually a subset of tumors that are characterized by the presence of the STAT6 mutations, but have highly indolent clinical cause, inguinal lymphadenopathy, large lymph nodes. So whether this makes clinically sense, I don't know, but biologically it would make sense in a subset of tumors. Yeah.
Okay, thank you. Juliette, you confirm me that we have no question in the chat?
Sorry, I was talking long and slowly and redundantly.
No.
I blame the jet lag. I blame the jet lag. So maybe people fell asleep during my presentation.
No, I don't think so.
So I disagree. Your talk was very good, and it was very stimulating. I have a question as I was thinking about your very nice story with this loss of function mutation in ARID1A. So at the beginning, when you set it up, you showed us the role of the microenvironment, and there have been, of course, a couple of different publications since then that have sort of described different types of microenvironments. And one thing that's difficult to study, but we should think about is like when we see individual mutations or mutational profiles, like what do we know about that interaction with the microenvironment? So for example, this particular mutation that you recently showed us, and you're showing this nice functional data, has it been associated with any particular microenvironment signature, or is that something that we don't yet know about that? Where are we with that analysis?
Yeah, so I have to admit we tried to look into that. Like you can use CyberSort X, different methods to infer the composition of the tumor microenvironment. This is something that is still ongoing. Our initial analyses we profiled almost 1,000 cases, but if it comes to the intersect of having the gene mutation data, having the gene expression data, having multispectral imaging, these numbers get low, right? So for EZH2 mutations, for instance, there was this association with specific signatures. For L1A mutations, we have not seen that yet.
Yeah. Okay.
But I think it requires larger numbers. So.
Yeah. One other question, going back to the beginning, is you showed us, some potentially actionable mutations now, right? So, I mean, one can make an argument if the data were available and I knew whether or not there was the presence of an EZH2 mutation, that might actually influence what chemotherapy I should select now. And I don't know if you agree with that statement or not, but one thing we get into a little bit is how much data is required before we, make these types of decisions, recognizing that oftentimes from a clinician's point of view, the decision between a bendamustine-based regimen and maybe something like R-CHOP is relatively arbitrary. So do you think there's enough information to say, well, if you knew, increasingly we do, next generation sequencing on patients as they get diagnosed. If we have that information, should we at least consider using that to select even upfront therapy, or do you think we have to have specific trials designed to answer that, or how should we think about this translation?
So I think a first approach would really be postdoc analyses. And this is what we're actually doing. So we teamed up with colleagues from Australia. There's validation cohorts from Canada. Because as you said, I wouldn't necessarily say it's arbitrary which patients receive bendamustine and which ones receive CHOP. But at least in our German guidelines, and I think this aligns with your guidelines as well, they are considered equally effective therapies. And then it's essentially depend on this side effect profiles, patient's choice, physician's choice, right? But we have patients, we know the genotypes, we have patients who receive bendamustine, we have patients who receive CHOP, and this is what we're doing right now. I think it requires independent validation. But I have to say that seeing this effect significant in both randomization arms of the gallium trial is meaningful, right?
Yep.
There is some
Plus,
question. Sorry.
Plus, we're looking into the biology of that, and I think we maybe not ready to be like we're putting together a manuscript right now. We've presented it last year at ASH. As I pointed out earlier, these EZH2 mutations, again, Christian Schneider has wonderfully shown this. One consequence of these EZH2 mutations is lower MHC expression on the cell surfaces. And like having an EZH2 inhibitor can reverse that phenotype. So in the lab, if we treat our EZH2 mutant cell lines with an EZH2 inhibitor, MHC expression goes up again. And then conjugate formation increases, T-cell mediated killing increases. So this can all be shown in the lab. Now, a funny thing is the same thing happens with CHOP. If we have sublethal doses of the CHOP regimen or its individual components, but doxorubicin is the most prominent compound, that also increases MHC expression. And I think that's the biologic explanation why these patients do better with CHOP-like regimens, specifically if they have an EZH2 mutation.
There is a question. We have one question. It's a question from Gabriel Brizou in Marseille.
Thank you.
Do you think we could develop a way to confidently identify patients sensitive to CHOP or BENDA at diagnosis? And how would you do this?
Yeah, I think that's a great question. So the confidently, the word confidently makes me hesitate. But I personally think analyses like that, so especially if we are addressing CHOP versus BENDA, we have patients who receive these regimens, right? I'm not saying that EZH2 mutation is the only determinant, but we have the patients, we have the cases, we have the analyses. I just think we have to do these association studies, right? In the end, everything needs to be validated, but especially if it comes down to CHOP versus BENDA, which is a question that I think there's also people from the industry joining, right? But I think it's fair to say that this is not the primary question that they will try to answer. But I think it's a wonderful question that academia can try to answer, right? These are two regimens that we give. And if we knew which patients benefit more from CHOP or BENDA, I think that would be a wonderful clinical tool. And I'm saying EZH2 mutation status and the 23 gene expression models both seem to interact with the efficacy of the chemotherapy backbone.
Yeah, I would actually say that I agree with what you just stated, and then say that those analyses, I really agree that it's going to need to be coming from academics, not only when we think about upfront therapy, but as all these newer therapies come. I mean, you touched on it, right? About the CAR T therapy coming or available, bispecifics coming, and one of the things we see consistently in the early studies when they become available is, the commercial sponsors are interested in telling us who does benefit. There's much less interest in telling us who doesn't benefit. And so what we see when there's subset, so as a general rule, if a study, maybe even if it's not as positive as we would like, we see subset analyses telling us, oh, but this patient population benefited. But if the study is positive, we almost never, at least I never see the analysis that says, by the way, this group of patients didn't benefit very much. I think we as a academic group are probably going to have to do those types of studies. Just if we do envision your title of biology driven therapy, we should not only think about who benefits, we should think about who is it that doesn't. It's possible, as you point out, there might be a group of patients with what you already showed that don't respond well to immunotherapy, and that's going to be very important. But there's not incentive enough for the commercial sponsors, certainly not at the beginning, to tell us that. So your work sort of sets the stage for other things we'll have to do once these things become available in large numbers.
Yeah, especially with these very expensive therapies, right? Like if we I fully agree. Yeah, this is important. Yeah.
Okay. I think everybody now is perhaps a little bit tired, but really I would like to thank you so much for this very, very interesting presentation with several aspects, more clinical part, more biological part. So it was really complete and very interesting, I think, for everybody. And I think it's probably time to Juliette is typing. Juliette is typing something, I don't know what, but probably it's time to close this very interesting talk session. I would like to thank you, Mark, to thank you, Oliver, and okay, thank you for this great talk, say Juliette. So it's perfect, and you could connect and see this talk again in replay session. So thank you very much. Bye bye.
Thank you so much for having me. Bye bye.
Bye bye.